Everyone to today's webinar of the International Eosinophil Society. Today we're going to have a webinar that's dedicated to eosinophil functions and how they relate to eosinophilic conditions and disorders. This webinar has been organized by one of our directors at large, Dr. Ueki. Next slide, please. And he has pulled together 3 presentations from some of our Asian researchers that are dedicating their work to eosinophilic functions. Next slide, please. This is just the mission statement of our society, and our webinars have been created in order to fulfill one of our major roles, which is to bring together scientists who are working in eosinophilic functions and eosinophilic disorders to exchange ideas and information about this particular cell type. If you want more information about our society, you can look at the link at the bottom of that slide. A few housekeeping issues. Throughout the presentations, all attendees will be muted. The chat feature will be disabled, but you can ask questions to the moderators anytime during the presentations using the question and answer function at the bottom of your computers. If you have any technical difficulties during the presentations, you can try to log off and log back on. That generally works, but we generally do not have many issues in that respect. And this webinar will be recorded, so you'll be able to view it or ask other people to view it after it is finished. It's my great pleasure to introduce our 2 moderators. First, Manali Mukherjee, that many of you know very well. She is our former trainee representative, now working at McMaster University as Assistant Professor of Medicine. And I also would like to introduce Dr. Ueki, who is the person who organized this meeting and who works in Japan in the Department of Internal Medicine and Clinical Laboratory Medicine at Akita University. So now, Dr. Ueki, you can introduce the speakers. Thank you.
So Manali, please.
Good morning. Thank you so much for the introduction. And it's a very good morning from Canada. And looking forward to a fantastic session that has been organized by Dr. Ueki. The first speaker for this series of talks in this morning's webinar. We'll start with Professor Hae-Sim Park, who's very well known for her studies, very elegant studies in asthma. She's from the Department of Allergy and Clinical Immunology from Jeju University, from the School of Medicine at South Korea. So Professor Park, please go ahead. We are very keen to hear your talk that will talk on the distinct role of eosinophils in severe asthma. So thank you and looking forward.
Thank you for kind introduction. Can you see my slide well? I
can't see your slide yet.
Yes.
We can't see your slide yet.
Yeah. Can you see my slide well?
We can, Professor Park. Thank you.
Thank you. Yeah. Let me start my talk. It is great honor to introduce a kind introduction, and I will update the recent data on the eosinophil in severe asthma. And my content today is that I will introduce the higher eosinophil count and the EEG forming eosinophil in severe asthma patients, and how EET may affect airway inflammation in asthmatic airway, particularly interacting with epithelial cells, and platelet aggregated eosinophil may have some role in this pathway. So we are very curious that whether any good biomarkers for representing EED forming eosinophil in severe asthma, and how to apply this knowledge in the management of a severe eosinophilic asthma. And we all know that type 2 eosinophilic inflammation in asthmatic airway, when eosinophils are more activated and they release EED forming eosinophil, they can enhance type 2 airway inflammation in asthmatic airway, and also enhance airway remodeling and finally leading to lung function decline and the poor clinical outcome. We all know that the degranulation in the EET forming cell set and the eosinophils are primed with the type 2 cytokine. They can activate it and increase the chemotaxis and transmigration and the cytokine release, and also degranulation products such as ECP and the EDN and NDP were released. And when atosis occurred in asthmatic airway, they have produced more prolonged and severe type 2 eosinophilic inflammations. So moving to the clinical aspect of severe asthma, and the eosinophil numbers is quite high in severe asthmatic patient than non-severe asthma, and lung function decline is noted in severe asthma. These two factors, higher eosinophil count in the low lung function, is a very important factor for predicting asthma exacerbation. In addition to eosinophil count, that we can see that the high eosinophil related markers such as serum EDN and the periosin level were higher in severe asthma, indicating that increased eosinophil count and the eosinophil activation markers are risk factor of asthma exacerbation in severe asthma. We published recently very elegant data in the long-term outcome of severe asthma. Patients with a severe asthma, they are suffering from frequent asthma exacerbation for a long-term period of asthmatic medication. They had maintained anti-asthmatic medication, including ICS and LABA in the tertiary hospital, but still they have asthma exacerbation. And also FEV1 was quite low, and that they progressively decline even though they have maintained the medication. And also eosinophil count was the baseline level was higher, and that they have a persistent eosinophilia even though maintain anti-asthmatic medication. Therefore, that we have to need a further intervention to prevent the lung function decline and also control and suppress persistent eosinophilia to overcome and to convert the prognosis of severe asthma. Moving to the EET forming in eosinophil, the previous study showing that the EET were found in asthmatic airway, and the EET can stimulate by IL-5 and anti-LPS stimulation. In severe hypereosinophilia syndrome in the skin tissue, they have shown that the EET forming eosinophil is noted. They may enhance eosinophilic inflammation in skin tissue. In addition, in the sinus mucosa of allergic sinusitis, and they found that it traps in the sinus mucosa, they enhance severe, persistent, and long-standing type 2 eosinophilic inflammation, indicating that EET may contribute to persistent and long-term eosinophilic inflammations. We first found that the EET forming eosinophil count in peripheral eosinophil in severe asthma compared to the non-severe asthma, we found that significantly higher EET forming eosinophils, which were positively correlated with the serum EDN level, and also negatively correlated between FEV1 and EET forming eosinophils. And also in the when they are treated by a steroid and antioxidant, they found that the steroid have little effect on EET forming eosinophil, indicating that steroid anti-inflammatory agent has a limited action on EET forming eosinophils. In addition to the eosinophils, we found that the EET may activate the airway epithelial cell and the IL-2 activation via releasing IL-33 and TSLP, finally leading to eosinophil activation. We named the EET epithelial cell, IC2, and eosinophil circuit. This circuit is enhancing type 2 eosinophilic inflammation. We replicated the EET forming cells in severe asthma were higher, and also IC2 cell number were higher, and the significant correlation between IC2 cells, EET forming eosinophils. And a type 2 cytokine, IL-13 and IL-33 and TSB level was significant in severe asthma. Here to find and confirm the role of the IC2 cells in animal model, when they incubated with the EET in mouse model, you can see that the increased airway hyporesponsiveness were noted in asthma, in asthmatic mouse that, and when they are pre-incubated with the IL-33 and TSLP, you can quite significant suppression of AHR and also suppression of type 2 cytokine in bronchoalveolar lavage level in IL-5 and IL-13 level. You can see this is a very interesting level when compared to between IL-33 and TSLP antibody. IL-33 antibody have great suppressive effect in this model. We may suggest that when compared to these two epithelial cell antibody in this model, that IL-33 may have a great suppression effect in this pathway. And moving to autoimmune response induced by ET, which was published by Canadian groups and Prof. Nayer's laboratory, and the ET and the NET may enhance autoimmune response in asthmatic airway. Previously, we published that increased IgG level to cytokeratin 18 and T level in asthmatic patients compared to the normal. And in patients with the severe asthma, we published the autoantibody responses in serum of asthmatic patients. And in addition, that in ET is incubated airway epithelial cell, we found that increased expression of cytokeratin 18 and 19 and the transglutaminase 2 and the alpha amylase here, which was not suppressed by dexamethasone treatment, indicating that the EET may enhance autoantigen expression in airway epithelial cell, which were not suppressed by dexamethasone treatment. So moving to the serum biomarkers representing EET levels in asthmatic airway, we compared the serum EDN level, and it was positively correlated with the EET forming eosinophil, and also serum EDN were correlated with the peripheral eosinophil count as well. So significantly higher EDN level in severe asthma, and the high EDN level may predict asthma exacerbations. In addition, in other asthmatic cohort, we mentioned that in asthmatic patients, in patients with uncontrolled asthma, significantly higher EDN level was noted, particularly in severe eosinophilic asthma, which were positively correlated with the blood eosinophil count as well, indicating that EDN may represent the severe eosinophilic inflammation in asthmatic patients. In addition to EDN level in blood, we also found that the high EDN level in sputum, which was correlated with the sputum eosinophil count, and also blood eosinophil count there. Sputum EDN level was significantly higher in severe asthma than non-severe asthma, indicating that in the eosinophil activation markers can work in asthmatic airway as well as blood level as well. Moving to the platelet low in asthmatic airway on eosinophil activation, and we measured the CD61 platelet eosinophil aggregated form in peripheral blood. You can see that patients with uncontrolled asthma significantly higher level of platelet adherent eosinophil numbers, which were correlated with the TIMP1 and the TGF-beta1 airway remodeling molecules. In addition, in severe asthma, in eosinophil, we found that increased expression of PTY10, which is a sharing marker between platelet and eosinophils. And also, we published the lyso-PS, which may induce ET formation in asthmatic airway, which was significantly higher in severe asthma than non-severe asthma. It is quite very interesting that PAF, platelet activation factors, they can also induce ET forming and the EDN release in severe asthma. And this finding may suggest it is not direct evidence, but platelet may have some role and the eosinophil degranulation and the ET formation in asthmatic airway, particularly in severe asthma. So in summary, that how eosinophils are more activated in severe asthma, we suggest that the EET may enhance type 2 inflammation via epithelial cell in the innate immune responses. And also they previously reported that the T responses release high periostin and TGF-beta1, which may be related with airway remodeling and the lung function decline. And also we published that in type 2 severe eosinophilic inflammation, surfactant protein was significantly lower. So these all process to enhance the eosinophilic inflammation, epithelial cells have key roles to mediate these type 2 inflammatory responses. Moving to the neutrophils in the eosinophil activation, we published in the asthmatic cohort that increased CD60 positive activated neutrophils were higher in patients with the uncontrolled status than controlled in the partially controlled status. In patients with the increased neutrophil count, that they have a low lung function and high symptom scores and high airway remodeling markers. So increased neutrophil count is related to poor clinical outcome and also related with the airway remodeling markers. We published in several years ago that in patients with a severe asthma, high neutrophil count and net forming cells were noted. It's like in the EET forming cells in eosinophil, neutrophil release net formation, which is related with the persistent airway inflammation. And when that is incubated the airway epithelial cell, you can see that they can activate the epithelial cell and release the cytokine, and also destruction of a tight junction in epithelial cells. And in addition to very interesting that the net activated eosinophil more in severe asthma than in non-severe asthma. So indicating that type 2 neutrophil and the net, they may enhance type 2 eosinophilic inflammation, particularly in severe asthma. We've recently published a very interesting molecule from airway epithelial cell, S11A9. They may increase net formation in severe asthma. Finally, they can enhance type 2 airway inflammation and the autoimmune responses. And the right panel showed that EET may induce neutrophil activation as well. And when they're incubated the airway epithelial cell, you can see the MPO and the S1A9 production from peripheral neutrophils as well. So therefore, we may an interaction between neutrophil net, enhance the eosinophil EET formation, and EET also enhance net formation, neutrophil, and the epithelial cell activation. So finally, to moving to the treatment part in the severe asthma, we have 5 kinds of type 2 biologics. And it is very interesting data published in the allergy. And in the severe eosinophilic asthma and the AERD, and the patient with the severe eosinophilic inflammation in cases of ABPA and eosinophilic CRS and EGPA, and the EET finding was noted in the lumen or tissues. In those cases that the IL-5 antibody and the IL-5 receptor antibody have limited actions, and also IL-4 receptor antibody have a great suppression effect in factors, indicating that in this finding, as well as the IL-5 eosinophil and the epithelial cell derived IL-4 and the IL-3 may have a target molecule in this pathway. This is an interesting study in our animal model as well. And when developed the EET-induced airway inflammation model in mouse, that in the we can see that the increased level of the IL-4 type 2 cytokine and also IL-33 and TSL3 in this pathway to suppress TSLP and IL-3 from epithelial inflammation. IL-5 antibody has a limited action, but IL-13 antibody have a great suppress effect in this pathway. So in this pathway, EET-induced epithelial cell inflammation in severe asthma, IL-5 have they can suppress eosinophilic inflammation, but not suppress airway hyperreactivity and the cytokine levels. Instead of that, IL-13 antibody may have a significant suppression effect there. However, I have also questioned that we do not have any biologics to suppress non-eosinophilic or EET-induced neutrophil activation, because neutrophil may also induce EET forming. EET forming also induce neutrophil activation. So we have to develop the new target to suppress this molecule as well. Finally, I may suggest my talk in that how we target the eosinophil and the EET forming epithelial cell interaction in severe asthma. And when the eosinophils activate, we also use the IL-5 and IL-5 receptor. They have enough suppressive effect to that. But when they more activated and release EET, they can affect epithelial cell. In this case, that IL-4 and IL-13 antibody or epithelial cell targeted antibody may have a great suppressive effect. But we do not have any answer about whether neutrophil may induce EET forming in this pathway, how we can treat and suppress this pathway. Finally, also introduce our recent data that we have developed a new anti-IL-4 receptor antibody, which was published in the 2 years ago. And also recently we developed IL-4/5 receptor antibody, which may have a very comparable effect with the penalisma, and also NK cell mediated cytotoxic effect as well. And we also hope that the new antibody, and we also evaluated the combined effect of these two biologics, IL-5 receptor and IL-4 receptors antibodies. But in conclusion of my talk, we mentioned that the E-phoning eosinophils have a very important role in severe asthma. They enhance type 2 airway inflammation. And also when ET is activated, they can induce epithelial cell and innate immune responses, and also direct activate the eosinophil as well. And when eosinophilic inflammation, we suggest that platelet activation may have some role and induce the ET-forming eosinophils. And also in the serum markers and the serum EDM may have some role for representing a severe asthma phenotype in the ET-forming eosinophil, although we need further relevant markers. And finally, in the treatment issue, that the epithelial targeting biologics may have a more beneficial effect to control ET-mediated airway inflammation than anti-eosinophilic biologics. Thank you very much for your attention.
Thank you so much, Professor Park. We have one question. We have enough time to take one question. I'm sure there might be a lot of you who want to ask Professor Park about the wonderful talk that she gave and had a lot of thought-provoking questions in our mind. So can I please have the first question?
Yes.
I can see Dr. Rufus has asked a question for you. She says, thank you for a very clear talk. Have you looked at charcoal-laden crystal in parallel to the eosinophil traps? So she's talking about the CLC crystals.
Oh, yeah, thank you very much for your question that we did not look at the charcoal-laden crystals as well. I think that the charcoal-laden crystal is more severe forms of the ET-forming eosinophil, but in vitro we look at the ET-forming cells, which was induced by IL-5 and LPS stimulation. That even though we observed the ET-forming eosinophil, but we could not confirm the charcoal-laden crystals.
I see. And another question, I'm sorry, I believe it's from, I can't read the name, it's in Japanese, well, I'm compromised there, but the individual asks you, says that it's a wonderful talk, and he or she says, you mentioned NET also activated eosinophilic inflammation, then anti-IL-5 may also be effective on neutrophilic asthma. Thank you. It's from Dr. Tae-Bum Kim from Korea. She's asking about the NETs and anti-IL-5.
Thank you very much for your question that yes, anti-IL-5 may have a little effect on the ET-forming eosinophil. And also when the NET induced eosinophilic infiltration, I think that the IL-5 has a little effect. They have no effect on the neutrophilic asthma.
Okay, thank you. And we have time for just another question. So our IES leadership, Professor Mark Rothenberg from Cincinnati, has written that, Dr. Park, thank you for an excellent talk. A couple of basic questions. Have you determined if eosinophil extracellular trap is composed of chromosomal or mitochondrial DNA or both. Do you think that the pathways and role of eosinophils in severe asthma also apply to other eosinophilic diseases like EGPA and eosinophilic esophagitis?
Thank you very much for your question. That we measured both type of the DNAs in there, and there were no significant difference. We compared the component of the EET and between severe and non-severe asthma. We found that both forms were working, and no significant difference in the composition of the EET. And the second question that thank you very much. I think that even though I did not observe whether this system is working in the other eosinophilic disease such as the EGPA, but I may speculate that this ET, eosinophilic and innate immune responses may work in the other type of severe eosinophilic disease.
Dr. Rothenberg again is asking, is the EET made of mitochondrial or chromosomal DNA? If you can just shed a little bit light on it, because there has been a kind of a debate on it for a while based on that.
Yes, yes.
So do you think it is made of the mitochondrial DNA or chromosomal DNA, or are you not in the space to answer that?
Still, we will look at and in detail, and I will give the answer. Thank you.
Are these eosinophils live or dead, Dr. Park?
Live cells. We collected the eosinophil from the severe eosinophilic patient, then we measured the EET forming eosinophils.
And is it you usually do it by flow cytometry, or is it by immunostaining?
Flow cytometry and also confocal microscope as well.
Nice. Thank you so much. Thank you so much. We have to go to the next talk. So I'd like to hand over to my co-moderator, Professor Ueki, to introduce the next talk and take care of the questions. Thank you.
Okay, so this is Dr. Ueki from Japan, Akita. So I would like to introduce Dr. Yosuke Kamide from, he's a chief physician of Department of Respiratory Medicine at Sagamihara National Hospital. You may know that Japanese government has set up 2 central hub hospitals in Japan, and Sagamihara National Hospital is one of them. Dr. Kamide, please start your talk.
Thank you for your introduction, Dr. Ueki. I'd like to share my screen.
Yeah.
Can everyone see my screen?
Yep.
Okay.
So we can see your next slide so maybe you can change your setting or something.
Oh,
I can see you very well.
Yeah, but I can see your next slide also. So
okay.
Yeah, perfect.
That's okay.
Yeah.
Sorry. Thank you for your introduction, Dr. Ueki, and good morning or good evening, participants. My name is Yosuke Kamide. Thank you for participating in IES. It's an honor to speak here today. I'm not a native speaker of English, so please go easy on me. I will be giving a talk on relationship between Galectin-10 and EGPA today. This is my COI. As you know, EGPA, eosinophilic granulomatosis with polyangiitis. EGPA is preceded by asthma and is a systemic vasculitis accompanied by eosinophilia. EGPA affects multiple and vital organs, including neuron, lung, kidney, or heart, and it results in malfunction of these organs. Therefore, urgent treatment is needed. Charcoal Riding crystals. Charcoal Riding crystals was discovered by Charcoal and Riding in 1853. Recently, charcoal Riding crystals are recognized as markers of eosinophilic inflammation. Galectin-10 is a protein abundant in the cytoplasms of eosinophils or basophils, but not in granule. Charcoal Riding crystals is formed by Galectin-10. Recently, charcoal Riding crystals were reported to lead type 2 inflammation. Galectin-10 itself does not cause inflammation in the respiratory tract, but charcoal Riding crystals produce inflammation. So we hypothesized that Galectin-10 is a marker of pathological eosinophilic inflammation in EGPA. Human eosinophils contains a large pool of Galectin-10 by a certain stimulation. By certain stimulation, cytoplasmic Galectin-10 is released extracellularly. This is in vitro study. We stimulated eosinophils using PMA, fixed them at 15 and 180 minutes, then immunostained for Galectin-10 and MVP. Galectin-10 is immunostained with green, and MVP is immunostained with red. In unstimulated cells, Galectin-10 and MVP were localized in cytoplasm and in granules, respectively. Stimulated cells and releasing net-like DNA. Net-like DNA is stimulated with immunostained with blue, while only stained with MVP, indicating that cytolytic loss of Galectin-10 was not accompanied by loss of granular content. Net-like DNA is called etosis and has become a hot topic in recent years. And eosinophils are induced etosis by PMA stimulation. To quantify the proteins released by stimulation, levels of LDH, Galectin-10, and EDN in culture medium were measured. It resulted in the release of 51% of total cellular LDH and 45% Galectin-10, but only 7% EDN. These results indicated that the release of intracellular Galectin-10 was closely associated with cytolysis. Galectin-10 from eosinophils is thought to be measured in the blood. So the purpose of this study was to examine whether the pathological eosinophilic inflammation of EGPA can be quantified by measuring Galectin-10. Here is a research method. Patients who were diagnosed as followed in Sagamihara National Hospitals after April 2000. Patients with EGPA, asthma, and healthy adult controls were included in the study. The study included cases over 20 years of age. EGPA were diagnosed by ACR criteria. And EGPA patients were divided into active or inactive patients. Active EGPA was characterized by increased eosinophil count, more than 10% eosinophils, or more than 1,000 eosinophil counts per microliter. And active vasculitis symptoms listed in the BVAS in at least one involved organ as histological, clinical, or laboratory data. Asthma was diagnosed by ATS criteria. Exclusion criterion was received antibody-based therapy. Then, serum protein were measured using ELISA kit. This figure shows the patient characteristics and laboratory data. There are 15 subjects in each group. There is no statistically significant difference in age, sex, BMI, and so on. Unsurprisingly, blood eosinophils and IgE are high in active EGPA group patients. The left hand, this graph is the levels of serum Galectin-10. This is EDM and ECP. As you can see, levels of Galectin-10, EDM, and ECP were significantly elevated in patients with active EGPA in comparison to remission EGPA, asthma, and healthy control. We then investigated whether the clinical symptoms in EGPA patients were associated with levels of Galectin-10 and granular proteins. The disease activity was characterized using BBAS. This is Galectin-10 and EDM ECP. As expected, there was a positive correlation between BBAS score and all shown serum eosinophil-derived proteins, including Galectin-10, EDM, and ECP. To exclude the possibility that blood eosinophil density affected these results, we compared serum concentrations of these proteins normalized to blood eosinophil count. This graph represents eosinophil-derived proteins divided by blood eosinophil count. Normalized levels of Galectin-10 was significantly elevated in patients with active EGPA in comparison to remission EGPA, asthma, or healthy control, but not of granular proteins like EDM or ECP was significantly elevated in patients with active EGPA. When serum concentrations were normalized by blood eosinophils, Galectin-10 was positively correlated with BBAS score, but granular proteins, EDN or ECP, showed a negative correlation. These results indicated that Galectin-10 is a unique biomarker for patients with EGPA. Since the IL-5 eosinophil axis plays a critical role in the pathogenesis of EGPA, we measured serum IL-5 in all subjects. As shown in the left-hand graph, serum IL-5 levels significantly increased in active EGPA patients compared with other subjects. In addition, a positive correlation between Galectin-10 and IL-5 was observed. We further assessed the relationship between IL-5 and eosinophil count, Galectin-10, EDN, and ECP in each group. R values were calculated with Spearman's rank correlation coefficient. Of note, a positive correlation was observed only with Galectin-10 in the active EGPA group. Recent findings reveal that lytic degranulation represents a process of active cell death. This is called etosis. Neutrophilic etosis is involved in the pathogenesis of ANCA-associated vasculitis. Therefore, it is expected that etosis is involved in EGPA mechanism, especially eosinophilic etosis. Galectin-10 and chalcolidin crystals are considered as a marker for etosis. So we studied affected tissues from patients with active EGPA to confirm this view. In the left picture, H&E staining showed the infiltration of intact eosinophils as well as chromatolytic cells and free eosinophil granules. In the middle picture, immunostaining of identical fields indicated the lytic eosinophils contains net-like citrullinated histone H3 and DNA stained with green. And citrullinated histone H3 stained lytic eosinophils were detected from patients with EGPA. The characteristic ultrastructural morphology of E. etosis was also confirmed by transmission electron microscopy. We did not observe eosinophils with apoptotic or necrotic morphology. Cutaneous histopathology of EGPA patients is presented. Picture on the left is control. Biopsy samples were immunostained for Galectin-10 and MVP. Intact eosinophils retained MVP and Galectin-10. This is pointed to by arrowhead. No cells were positively stained for cytoplasmic Galectin-10 alone. Whereas lytic cells and free granules were only positive for MVP. This is pointed by arrow. Separate, distinct, and focal extracellular punctate MVP and Galectin-10 stainings are indicative of free extracellular granules and vesicles, respectively. Cytolytic eosinophils were detected and were often associated with small punctate Galectin-10 labeling, which were probably eosinophil extracellular vesicles released from E-etotic cells. These pathologic findings are found in not only skin, but other organs like gastric mucosa or lung. These are figures from one patient. Cytolytic eosinophils were observed in 21 samples from 16 patients. Intact eosinophils retained MVP and Galectin-10, whereas lytic cells and free granules were only positive for MVP. So in summary, levels of Galectin-10 was significantly elevated in patients with active EGPA in comparison to other groups. To exclude the possibility that blood eosinophil density affected these results, we compared serum concentrations of these proteins normalized to blood eosinophil count. Normalized levels of Galectin-10, but not granule proteins, were significantly elevated in patients with active EGPA. Serum IL-5 levels significantly increased in active EGPA patients compared with other subjects. In addition, a positive correlation between Galectin-10 and IL-5 was observed. In pathological examination, citrullinated histone H3 stained lytic eosinophils were detected. The characteristic ultrastructural morphology of E-etosis was also confirmed using TEM. And cytolytic eosinophils were detected and were often associated with small punctate Galectin-10 labeling. In active EGPA, concentration of Galectin-10, EDN, and EGP were elevated. However, normalized levels of Galectin-10, but not granule proteins, were significantly elevated. We demonstrated that levels of these proteins are correlated with blood eosinophil counts or BPAS. It is considered that these results were reflected that blood eosinophil count is not always high in patients with high granule proteins, but levels of Galectin-10 were high in almost all patients with high blood eosinophil counts. positive correlation between Galectin-10 and IL-5 was observed, indicating that IL-5 should cause eosinophilic inflammation by the mechanism different from eosinophilia antigranulation. Our data indicated that eosinophils infiltrating into various tissues underwent E-etosis in EGPA patients. In conclusion, thanks for your attention.
Thank you, Dr. Kamide. And we have several questions, and we have a few time to Q&A session. Our first question is Dr. Manali asking, did you see a difference in Galectin-10 levels based on their anchor status?
That's a good question. We checked anchor. However, the presence or absence of an anchor does not affect Galectin-10 levels.
Okay, thank you. So next question is from Florence. Did you observe any relation between Galectin-10 and neutrophil counts or MPO levels? What are difference between EDN and Galectin-10 levels in eosinophilic inflammation? This is from Professor Park. Sorry. So Galectin-10 and neutrophil count.
Yes. Galectin-10 and neutrophil count.
Neutrophil count.
Okay.
We have another question. Platelet count. What about platelet count?
We've never done that about neutrophil, so I'd like to try.
Yeah, well, we should know. We don't check the platelet count and Galectin-10 levels. We don't see any association between Galectin-10 and platelet, right?
Yes, platelet. Yes, that's an important question. But so platelets related to we don't try to calculate.
Yeah,
However,
it's very diverse. Yeah, I think. So, yeah, maybe the platelet levels were also statistically significantly different among patients subgroups, especially after the preceding talking showing that platelets can favor eosinophil activation and E-etosis. So yeah, that's important point. So another question is, Taebam came from Korea, saying wonderful talk. How severe asthma patients were enrolled in this study? Because when I measured the levels of EDN, the levels of EGPA was lower than that of severe asthma. Yeah, so
we enrolled severe asthma using genus asthma criteria. And
the severity.
Yes.
Was
severity equality severity check, and so we enrolled severe asthma using GINA criteria.
So, but not it's stable condition, right?
Yes. Yes. Severe asthma and uncontrolled asthma criteria. So, yes. For example, we use the GINA step equal to active EGP and asthma. So GINA step 3, 4, 5 is enrolled in asthma patients.
Okay. So such patient didn't have an increased level of Galectin-10 in our study, right?
Yes.
Yeah. Okay. So Professor Rosenbaum asking, can you explain why EDN is not released from PMA-treated eosinophils as you showed that the cells are dead as assessed by LDH? Related, what is the mechanism whereby CLC is especially released by E-etosis? Maybe I can answer the question. I think so. EDN is inside of the granules. So we corrected the supernatant with 1000 G, which is only separate, which can separate the granular intact granules from medium. So that's a reason we cannot detect EDN by from the E-etosis cells. And what is the mechanism whereby CLC is especially released from by E-etosis? Yeah. So again, the from in the process of E-etosis, the granule is almost all granules are intact. So that's why so we can detect Galectin-10 levels in serum supernatant. So it might be time to so any comments on Dr. Kamide? Because I answered.
Thank you.
Okay.
Yeah, comment.
So, okay, so that it seems to be time is running. So, thank you, Dr. Kamide, and we will move on next speaker. So, Manali.
Hi.
Hi. Thank you.
Thank you so much, Professor Yuki. Okay, our final talk for this morning's Eosinophil webinar from International Eosinophil Society. We'd like to welcome Professor Jun Miyata from the Division of Infectious Diseases and Respiratory Medicine, Department of Internal Medicine from the National Defense Medical College. Dr. Miata will be speaking to us about dysregulated fatty acid metabolism of human eosinophils in allergic diseases. We're really keen to hear a very unique topic. The floor is all yours, Dr. Miata.
Thank you for your introduction. Can you hear me?
Yes, we can. Please, if you can share your slides.
Okay. Share my PowerPoint slide on the web. Can you see my slides?
We can indeed. If you can go into the presentation mode, please. There you go. Perfect.
Okay. And also, I'd like to thank Dr. Ueki to give an opportunity to make a presentation here. This is a great honor to speak to you today. So in this presentation, I'd like to focus on dysregulated fatty acid metabolism from human eosinophils in allergic diseases. So in this slide, I disclose my COA relationship. So my presentation consists of 3 parts. First, I'd like to talk about the severe eosinophilic asthma, its phenotype, and its relationship with eosinophilic rhinosinusitis. And next, I'm going to explain about our basic research using about dysregulated fatty acid metabolism of nasal polyp-derived eosinophils. We performed multi-omics analysis and used the bioinformatic method. So today's talk mainly focus on these results. And lastly, I'll briefly talk about impaired synthesis of specialized pro-resolving mediators in severe asthma. So I like to start by talking about the severe eosinophilic asthma. Asthma is characterized by chronic airway inflammation, airway hyperresponsiveness, airway remodeling, airway obstruction, and respiratory symptoms. There is an increasing number of patients worldwide, and its morbidity is very high. Eosinophils play central roles in types of inflammation. Eosinophils are much potent, much functional granulocytes capable of releasing various types of chemokines, cytokines, and lipid mediators, and function as main effector cells. So it is very important to understand the cellular properties of eosinophils in inflamed tissue and in disease state. So recent study demonstrated that asthma can be classified into several phenotypes based on patient characteristics. Onset age is very important in this point. Adult onset asthma is characterized by eosinophilic inflammation and chronic rhinosinusitis, and NSAID exacerbated respiratory disease often occur in adult onset asthmatic patients. So we focus on the disease in the red square. This slide shows a relationship among severe asthma, eosinophilic chronic rhinosinusitis, and NAD. 5 to 10% of asthmatic patients are refractory to standard therapy, which is defined as severe asthma. Patients with severe asthma often have NSAID intolerance and more frequently have chronic rhinosinusitis and nasal polyp. So we planned research strategy to using nasal polyp taken from patients with chronic rhinosinusitis. So I'll move on to the second part about fatty acid metabolism of nasal polyp eosinophils. This slide shows synthetic pathway of arachidonic acid-derived lipid mediators. Arachidonic acid is metabolized into many types of lipid mediators via multiple enzymatic reactions, including cyclooxygenase and lipoxygenase. These lipid mediators have broad activities, including pro-allergic effects of cysteine leukotrienes and prostaglandin D2. In contrast, another class of mediators, lipoxins, have anti-inflammatory and pro-resolving effects. In recent studies, many types of anti-inflammatory lipid mediators are discovered and characterized. These mediators are called specialized pro-resolving mediators, SPMs. SPMs are synthesized partly from arachidonic acid, lipoxins, and mainly from omega-3 fatty acid, DHA, including protectin and resolvins. These mediators inhibit PMN infiltration and enhance apoptotic cell clearance and promote the resolution of inflammation. So our lipidomic analysis of eosinophils taken from healthy subjects reveals that eosinophils synthesize large amounts of leukotriene C4 and thromboxane B2. SPMs are not detected, but adding DHA to culture medium dramatically changed this profile. The amounts of inflammatory mediator were reduced, and a specific type of SPMs are produced, especially protectin D1. So these results indicated that eosinophils have potent biosynthetic capacity of lipid mediators. So we are interested in regulatory mechanism of this system. So in this slide shows research strategy using multi-omics analysis of human eosinophils. Multi-layer omics are composed of transcriptomics, proteomics, and lipidomics. Data is analyzed by bioinformatic method. We applied this protocol to blood eosinophils and nasal polyp eosinophils taken from patients with severe asthma and chronic rhinosinusitis. First, I show data set of blood eosinophils from healthy subject and nasal polyp eosinophils. This slide shows the protocol of an omics sample preparation from human eosinophils. We use the cell sorter to isolate human eosinophils in a negative selection method. Granulocytes have large quantities of proteins and RNAs to decompose RNA and proteins easily. So we developed a new protocol to isolate intact RNA and proteins by using trizol. And in lipidomics, solid phase extraction was performed for oxidized lipid extraction, and targeted lipid mediators are quantitatively measured by L-C mass spectrometry. So we performed morphological analysis of nasal polyp eosinophils by using microscope and flow cytometry. Nasal polyp eosinophils are larger than that of blood eosinophils. That confirmed by measuring area, circumference, and diameter. Also, in flow cytometry-based analysis, we observed higher FSC and lower SSC in nasal polyp eosinophils. We investigated the expressions of surface markers, the level of CD69 and an activation marker where eosinophils are most elevated in nasal polyp eosinophils. And we also observed lower levels of Shigeharu eosinophil specific markers and CCL3 eosinophil specific chemokine receptor. So we performed the lipidomic analysis. These results revealed dysregulated fatty acid metabolism in nasal polyp eosinophils. We observed this impaired synthesis of 15 lipoxygenase-derived mediators and cyclooxygenase-derived mediators. And among 5 lipoxygenase-derived mediators, selective enhancement of LTD4 synthesis was observed. So this slide shows the synthetic pathway for sustained leukotriene metabolism. Production of leukotriene D4 is mediated via 2 types of enzymes, gamma glutamate transferase and G-peptidase. So we measured the expression of 2 types of enzymes by proteomic analysis. In proteomic analysis, in total, 2,298 molecules are measured. And compared to blood eosinophils, nasal polyp eosinophils had 100 upregulated molecules and 150 downregulated molecules. But these expression patterns were quite similar between both groups. Major proteins contained in eosinophils are eosinophil-specific granules. Amounts of these proteins are equivalent between both groups, blood eosinophils and nasal polyp eosinophils. But PLG2, called major basic protein, slightly increased in nasal polyp eosinophils. So this slide shows a top 10 list of upregulated and downregulated protein in nasal polyp eosinophils. Among these proteins, gamma glutamate transferase 5 most elevated in nasal polyp eosinophils. And among lipid converting enzymes, the expression of D-peptidase 2 decreased in nasal polyp eosinophils, 80% decreased. So this slide shows, so gamma glutamate transferase and G-peptidase have several isotypes. So this slide shows the expression of these isotypes. So we found that selective expression of GGT5 and G-peptidase 2 in eosinophils. So this slide summarizes characteristic changes of fatty acid metabolism-related enzymes in nasal polyp eosinophils. In addition to the changes of GGT5 and the D-peptidase 2, we observed increased expression of 15-lipoxygenase and decreased expression of 15-lipoxygenase and increased expression of ALX5-AP flap in nasal polyp eosinophils. So again, we show synthetic pathway in system leukotriene metabolism, and these changes in nasal polyp eosinophils increase the conversion of leukotriene C4 into leukotriene D4. However, inhibit the conversion of leukotriene D4 into leukotriene E4. So this might result in selective accumulation of leukotriene D4. So we want to know the upstream regulators of these changes. So we performed the transcriptomic analysis. In total, 90,529 molecules, protein coding genes were measured. And the expression pattern quite different between both groups. And too many genes upregulated or downregulated in nasal polyp eosinophils. So to comprehensively understand the nature of nasal polyp eosinophils, we performed the principal component analysis of transcriptomic data. This slide shows nasal polyp eosinophils totally different from peripheral blood eosinophils in PC1 component, gene expression pattern of PC1 component. And the GGT5 and the G-peptidase 2 are major contributor to PC1 component. Next, we performed the pathway analysis of transcriptomic data. We identified inflammation or microbe-related signaling pathway, identified them as candidate of upstream regulators in activation process, and also identified type 2 cytokines, GM-CSF, IL-4 and IL-5, or pattern recognition receptors, TLR2 and NOD2, as candidate molecules with high activation Z-scores. So to examine the role of type 2 cytokines and pattern recognition receptors in the formation of nasal polyp eosinophils, we performed in vitro studies using blood eosinophils from healthy subjects. We stimulated eosinophils with various cytokines. Among these cytokines, IL-4, IL-5, and GM-CSF upregulated GGT5 expressions. In contrast, G-peptidase 2 downregulation was elicited by stimulation with IL-5 and GM-CSF, not IL-4. So we used IL-5 and GM-CSF. IL-5 and GM-CSF stimulation for 3 days induced nasal polyp-like fatty acid metabolism. We observed impaired synthesis of 15-lipoxygenase or cyclooxygenase-derived mediators, and selective enhancement of LTD4 synthesis. And also, pattern recognition receptors might be important in the pathogenesis of nasal polyp eosinophils. Among pattern recognition receptors, NOD2 and TLR2 mimic interaction with Staphylococcus aureus. This pathogen is important and common in nasal cavity and release toxins and elicit inflammatory responses. So these agonists synergistically upregulate GGT5 expressions and downregulated D-peptidase 2. Similar to IL-5 and GM-CSF stimulation, these agonists induce nasal polyp eosinophil-like fatty acid metabolism. We observed impaired synthesis of these mediators and selective enhancement of LTD4 synthesis. So in summary of this section, we identified dysregulated fatty acid metabolism in nasal polyp-derived eosinophils. These eosinophils are presented with inflammatory phenotypes characterized by GGT5 upregulation and LTD4 overproduction. These changes could be induced by interaction with type 2 cytokine producing cells or microorganisms. So these findings provide new therapeutic targets in this disease. So lastly, I move on to the third part about impaired synthesis of SPMs in severe asthma. So again, we performed the lipidemic analysis of human eosinophils, blood eosinophils from healthy subjects and severe asthmatics. Amounts of arachidonic acid and 5-lipoxygenase-derived mediators released from eosinophils are similar between both groups. However, selective impairment of 15-lipoxygenase-derived mediators are observed, including lipoxin A4 in severe asthma. So recent studies demonstrated impaired synthesis of 15-lipoxy-derived lipid mediators that are observed in many clinical samples, and those taken from patients with severe asthma, NAD, and acute exacerbation. And we and other groups identified diminishing synthetic capacity of selective cell types, including eosinophils and alveolar macrophages. So eosinophils in severe asthma and eosinophilic chronic rhinosinusitis had diminished synthetic capacity of 15-lipoxygenase and SPMs. So this phenomena might result in excessive and unresolved inflammation in severe asthma. So eosinophils synthesize SPMs from DHA and eicosapentaenoic acid, EPA. These molecules have been discovered and characterized. These molecules inhibit allergic inflammation via suppression of mast cells and dendritic cells. And these molecules, we demonstrated that these molecules promoted the resolution of over-induced airway inflammation via suppression of L-taxin production in airway epithelial cells. And also, we demonstrated that DHA-derived SPMs suppress IL-C2 activations. Systemic deficiency of 12 and 15 lipoxygenase caused enhancement of IL-33 induced airway inflammation and impaired synthetic pathway of 12-15 lipoxygenase-derived mediators and SPMs. And we identified specific SPMs, protectin DX, resolvin D1, and malacin, suppressed IL-C2 activation, IL-5 production, and cell proliferation. These results were obtained by lipid screening by using IL-C2 in vitro. So these results indicated that dysregulation of 15-lipoxygenase possibly cause enhancement of airway inflammation in severe asthma. So that's all I have to say. I'd like to thank all my collaborator and colleagues. Thank you for your attention.
Thank you so much, Dr. Miyata. It was very interesting and very unique as well. Beautifully done work as well, and the presentation was very clear. I'd like to open the floor for questions. Please, if you may raise your hand or type in a question directly that I can ask. So I'm going to go with the first question since I don't have any. So Dr. Miyata, you showed the difference between the different markers in the peripheral eosinophils and the eosinophils from the nasal polyps. We know that a number of patients have comorbidities with severe asthma. Did any of these patients have severe eosinophilic asthma?
Patient with nasal polyp?
Yes.
Or eosinophilic rhinosinusitis? No, all patients with nasal polyp have do not have severe asthma, but the rate is high. Severe asthmatic patients often have comorbid.
Correct.
Nasal polyp.
That's exactly why I'm asking. So any one of them had asthma amongst the patients that you recruited?
Included? Okay. Included patients have one or two patients have severe asthma, but not all patients.
That's interesting because this might be very similar if we look at maybe eosinophils from the airways, and they may have a dysregulated lipid profile as well. How do you think the corticosteroids may affect? Because correct me if I'm wrong, patients with nasal polyps are also treated with corticosteroids, and so are severe asthmatics. How do you think corticosteroids would affect this lipid profile?
Thank you for your question. Enrolled severe asthmatic patients, 2 in 5 patients have corticosteroids, but have dysregulated fatty acid metabolism. So, but we checked corticosteroid treatment diminished cellular changes of eosinophils and improved dysregulated fatty acid metabolism. So some patients are refractory to corticosteroid, and maybe possibly via GR expression, downregulated GR expression, or as a mechanism.
Right. And I have a question from Sanjay Ramakrishnan here, who asks you, how does this relate to aspirin sensitivity?
Thank you for your great question. Not all patients have aspirin intolerance, but aspirin intolerance is closely related with chronic rhinosinusitis. So this our findings suggested that this dysregulated fatty acid metabolism is observed in patients with aspirin intolerance.
Okay, I'm moving to the next question from, Sanjay again says, would aspirin intolerant patients make a unique model to study? Do you think the aspirin intolerant patients will make a unique model to study? You intend to study that population?
Thank you for your kind question. And yes, adding aspirin to eosinophils taken from patients with aspirin intolerance produce systemic leukotrienes. But I think mechanism remains unknown. So this is very unique and interesting model to study. Thank you.
Right. And my next question is from Dr. Amy Cleon, who's from our IES leadership. She's asking you, have you performed similar studies on patients who are on anti-IL-5 therapy to see if their eosinophil profiles are reversed? So it's very much complementary, goes hand in hand with the question with corticosteroids. So how do you think the lipid profile in eosinophils changes with anti-IL-5 therapy?
Thank you for a great question. I plan to do it So this is very important. Recent studies published on ELG, patients with anti-IL-5 therapy have normal eosinophils gene expression profiles compared with healthy eosinophils from healthy subjects. So I think IL-5 antibody treatment reverse this profile, possibly. Thank you.
Great. I have two questions on SPMs. So Professor Park here, our first speaker for the series, is asking you, is there any serum biomarker for representing SPMs? And Dr. Francesca Levi-Schaefer is asking if you notice any effects of eosinophil-derived SPMs on cells other than ILC2s. So two questions on SPM.
Yeah. Thank you very much for your questions. And it is very difficult to measure specific SPMs. Fatty acid derived metabolites have similar texture or component. So ELISA is not specific for each metabolite. So we use the LC mass spectrometry, but it is difficult to measure SPMs. And derived SPMs on cells other than LCs.
Yes.
I know that SPMs inhibit mast cell activation or dendritic cell activations, and neutrophil infiltration into inflamed tissue are also inhibited via SPMs. So SPMs mainly have inhibitory effects on type 1 inflammation, but slightly on type 2 inflammation. This is the immature speculation. Thank you.
Thank you, Dr. Miyata. I would also say that you did show a really cool validated method of performing lipidomics with eosinophils. And for all of us who love eosinophils, we also hate it when methods don't work out with this cell. It's very difficult. So usually we do have an Ask an Eosinophil expert session in our in-person meetings. So we would definitely be asking you to present in one of the Ask, you should have one poster or so where you can share with our, with our members how more clearly how you did that method and how you validated that, because it's always something we want to learn on how to deal with these cells better, whether we are trying to get the proteins out, RNA out, or looking at the lipid profile. So thank you so much for speaking to us today. And I believe there are no other questions so far. And I'll pass it over to our president, Dr. Rufos, and close this session. Thank you.
Thank you very much.
So I would also like to thank everyone. I would like to thank in particular Dr. Ueki for pulling together the program. We had some very interesting presentations on unique eosinophil mediators and their presence, their dysregulated presence in various disorders were well highlighted by today's speakers. So thank you to Dr. Ueki for proposing the program and for moderating the session together with Manali Mukherjee. I would also like to, if you just go on to the next slide, please, announce our next meetings, the future webinars. They will be held in November and December. As you can see, we're continuing with eosinophil biology in November, discussing eosinophils and metabolic health and disease. And in December, there will be a special session dedicated to eosinophilic gastrointestinal disorders. Please note that these sessions will be organized at the same time as usual. Today's session was an exception, and it was to be able to span the different time zones of all the people involved in this particular webinar, but we will be going back to our 9:30 a.m. Eastern Standard Time for the following sessions. We will also have a nice session planned at the end of January. This will be a joint webinar with the European Mast Cell and Basophil Research Network. So this is going to be very interesting to be able to show the different interactions between these different cell types and how this also relates to disease. Next slide, please. And then once again, here's the link to our website where you can find more about the upcoming programs, but also a series of links to have the recordings to all the webinars that have been programmed so far during 2021. I thank you all for attending this session, and I hope to see you next month.